Hi, hello, namaste. This is Race tutorial, your Srinivas. Today, I'm going to discuss a wonderful topic like what is that? Molecular basis of inheritance. In that botany IPE, 5 questions are there. In that starting 3 questions are very important. I'm guessing any one question guaranteed will come. The remaining 2 questions also I'm going to give. So, let's start. The first question is like what is that? Please please subscribe my channel. Let's start by topic. Like what is that first question? How many types of RNA polymerases exist in cells? Write their names and function. This is the question. So answer is like what is that? You catch it. Here, how many polymerases is there? What are the names and what is the useful of function? There are asking questions. So here, very simplified. Race C tutorial is nothing but very psychological clue and easily identified clues are there. So catch up students. So here, three RNA polymerases are there. You catch out this. Here, three RNA polymerases. What are they, sir? Just here you check out explanations. This side, 1, 2, 3. Three types of RNA polymerases are there. And here, how many means? 3 or catch. So it is a transcript like what is that? R, RNA, M, RNA, tRNA. Ribosomal RNA, messenger RNA, transfer RNA. RMT you will catch. That's very simple. Here M is there mean headmaster you will catch. HM sir, HN RNA. HN means heterogeneous nuclear RNA. Here tRNA, here ST% headmaster sir is there. So STHM finish answer. S means srRNA and another one is snRNA small nuclear RNA. Sir, how sir you catch out? This is the first one is there. First one you check out from here. See 1. So here check out 1 rRNA. What are these sir? 28 years less 10, 18 years, next 5.8 years. Next, RNA polymerase 2. It is transcript the precursor of mRNA is there. See here, M and another one is hnRNA, heterogeneous nuclear RNA. Next, one more, 3 is there. Here, tRNA, check out 3. This is the tRNA and srRNA and SN small nuclear RNA. So here, ST and H headmaster finish this answer. Come on students, take screenshot of this answer and along with explanation easily with that. Finish this answer. You will get 4 marks, 4 by 4 marks. How many types are there? 3. What are they? 1, 2, 3. RMT, headmaster and that's it. SN, ST. Next, the second question. Write the important features of genetic code. So genetic code, what type of importance is there? Let's we are going to discuss it. It is understanding purpose also very simplified code I am going to use. You know, in Andhra Pradesh, which party is on? Telangana party. So Telangana party, TDP party, USI. This is one of the code. Finish this answer. How sir? How it is possible to check out? I mentioned from here, red mark. I put T D P U S I. Finish this answer. So the silent features of genetic code in that the first code I am going to say first point. The codon is a triplet. What's a triplet? We know you will take anyone. So triplet is present, codon is triplet. Total how many codons are there? T represent like what is that sir? Triplet only. T represent like what is that? Triplet. So total how many codons are there sir? We know 64 codons are there. In that 61 codons codes with 20 amino acids are there. So 64 codons in the 61 codons are codes with 20 amino acids. But 3 codons does not code anything, any amino acid. So that is called as what we'll call, sir, stop codons are the, what we'll call stop codon. So that's why here I clearly mentioned the codon is triplet. Total here 61 codon out of 64, 61 codon code for 20 amino acids. But 3 codons do not code anything, any amino acid. Hence it is called like what is that? Stop codon. So stop codon's definition is a 2 marks question, friends. This is a stop codons are these are 2 marks question. What are the stop codons? UAA. Here very clearly I am mentioned to UAA, UAG, UGA. So these are the stop codon 2 marks question along with 4 marks also there. Just check out T for triplet. This is the first one completed. Second one is D. D mean what, sir? Degenerate. That's it. D mean degenerate. In bracket point, if you want interest, you will write. Otherwise, it is useful in only for amino, just NEET examination. So some amino acids are coded by more than one codon. So just otherwise, if you want interest, you will write it. Otherwise, just you write from here up to here only. The codon is the code is degenerate. Next one is like what is that? P is there. P mean like what is that? Punctuations are there. Punctuation mean what, sir? Here I mentioned like what is that stop codon, but in that comma, full stop I am not mentioned. So that's why what happened here? There is no punctuation in the middle of the codons are there. So P, D mean your catch, degenerate. P mean no punctuation. Right. I think that catch. Next U is there. U mean like what is that? Decompleted, T completed. U. U mean what, sir? Universal. So what's a universal? U mean universal. You go anyway, the same codon will follow. Universal. U is represent like what is that? Phenylalanine symbol. So you can catch up. Next one is the code is the specific and unambiguous. So S represent like what is that? Specific. Just a specific you will catch up. Okay. The code is specific and unambiguous. Unambiguous means doubtless. There is no doubt in the middle of the codon or code. Next, sixth one, I mean represent like what is that? Initiator codon. Initiator means starting. That is the identification. A, ag, agu. In Telugu, what we are saying, if you are going anywhere, A, agu, stop there, agu, A-U-G, ag. This is 2 marks question you can. So ag has a dual function. One is a code for methionine and another one is act as initiator codon. So this is if you know the code like what is that? TDP-USA, you can catch this complete answer. So T, triplet, degenerate, no punctuation, universal and specific and initiator codon. Next, the third question. What are the differences between DNA and RNA? So it is also very simplified answer I will give you. One small code I will give you. So just easily will get that answer. So let's see here. What is the difference between DNA and RNA? Very clearly mentioned. Take screenshot, friends. So DNA mean we know deoxyribonucleic acid. RNA is a ribonucleic acid. What type of differences is there? So this is the understanding purpose. DNA square. I'm giving to you. What's up? DNA D here. Deoxyribose sugar. So DNA deoxyribose sugar. But here RNA ribose sugar is present. This is the first point completed. Deoxyribose sugar. Second, one more square is given for 2 mean here it consists of 2 strands of polynucleotide cells. So we know like what is that DNA structure is 2 polynucleotide strands are 1 and 2. But here RNA is only one type. Here 2 double DNA is a deoxy, another one is a double strand. Here 2 strands are mentioned. So that is the you will catch. 2 points completed, sir. Finish? Yes. Next, one more point. Here, MPRG. Finish this answer. What's the MPRG? So, M represent like what is that, sir? M. Metabolically, DNA is only one type is present. Only DNA, only one type. But here, metabolically, RNA, 3 types are there. What are these, sir? rRNA, mRNA, tRNA. So that's the 3 types are there. So M represent like what is that? Metabolically. That's it. Okay. I think the fourth one is like what is that? P. P means protein synthesis. So DNA that does not participate in a protein synthesis indirectly. But here, what about this RNA is directly participated in protein synthesis, transcription is completing after translation process. Like this total which are participated like what is that RNA. So in that RNA polymerase is very, very important. That we are going to discuss in NEET level. It is only for IPE, no? So simply I am going to say RNA is directly participated in protein synthesis. DNA does not participate. So P, protein synthesis. That's it. So protein synthesis not participated. Next, fifth one. R, DNA is a replicative project. How, sir? We know semi-conservative method. One mother DNA which may produce the 2 daughter DNA are there. That is by the helping of DNA semi-conservative method replication is shown. So simply here DNA is replicate. But here RNA does not replicate. Finish. R means completed. Here one more like what is that? G is there. G means like what is that? Genetic material. So here DNA is genetic materials are there, but RNA is not a genetic material. We told in NEET level like what is that? Hershey says one of the 2 marks question of important. DNA is genetic material. Finally concluded by who? Hershey and Chase. This is a 6-point part. Are you catch it? So MPRG, that's it. Sir, one more extra point I'm going to read, sir, mean just 6 points is enough to you. So if you want interest, more enthusiasm, you read it like what is that is important in NEET level. What are these are? Pyrimidines are they? What type of pyrimid? Cytosine and thymine is there. But what about this RNA? Here thymine is absent at the places like what is that? Uracil is there. So CT here, there CU. That's its answer. Next, fourth question. The start fourth question is draw the schematic and diagrammatic presentation of the lac operon. So diagrams only there are asked. So just diagrammatic only. How to you will draw? Easily choose everything. So if you know the code, like what is that? PIPO ZYA. This is one of the easily identified code. You substitute these boxes PIPO ZYA. What's a ZYA is a structural J. So you write it this. Here also same like that only you will write it. This is the in the absence of inducer is one box. Here presence of inducer. So you separated boxes and draw the diagram and substitute this P, PO, ZY. That's it. So very simple, no? Very simplify of this P, PO, ZYA. But here regulator gene is there, but it may produce like what is that, a repressor mRNA. Just you write it like this, and which is produce repressor. So here what happened, in the absence of inducer you will write it. In the absence of inducer means there is no presence of lactose. So what happens, this R repressor is bound with O mean operator. So repressor is bound with operator to stop the transcription process. So that's to prevent the RNA polymerase from transcribe the operand. So prevent means stop the operand. So that's it. This is a simple diagram now. If we know this diagram, we can draw one more also. Same similar is this. Check out. Right? I think you catch it now. Here in the absence of inducer you will catch. Next in the presence of inducer. Same. I'm written P4ZY. So in the presence of inducer mean what happens, sir? Lactose we are using. Lactose is nothing but what will come? Inducer. So inducer I use mean what happened? At that time the repressor is inactive is present. So that's the inactive repressor. So simple. Inactive repressor. So it is not bound with operator. So then what happened? Transcription process, translation process started. So like transcription I here I'm written, translation here written, and just mRNA written. Why? Because this is the double stranded DNA, transcription process after mRNA is produced, then after translation process is started. So you check. Then after this structural gene ZYA which is produced another gene. How, sir? BP tablet you use. So how, sir, here BP tablet, BP tablet you use BP tablet. What, sir? B galactose permease trans acetylase. So this is the identification code BP tablet. BPT, that's it. So people divided by A, BPT. If you know this code, we can write everything. BPT. Come on, take screenshots, my dear students. So first you write it, draw this diagram. Second, and after what happens like this process, it is bent like this to stop the transcription process. Then after this started means inducer is used. That is nothing but presence of inducer. So diagram, finish the diagram. It's very easy now. I think you took screenshot. You read this one only. No need to read any other reference books. Directly you draw this diagram. What I am given notes, you will should follow it like that only. You will prepare like that only. Right. Next, fifth question. Write the briefly on nucleosome. Very simply by Andy. Nucleosome explanation there are asked. Very shortcut method I will tell you. Just concentrate on about. So in eukaryote, this is a blue color mark is there. That is the double stranded DNA. Here red mark is there. That is called histone octamer. So in eukaryote, dsDNA wrapped around the histone octamer. That is called as what will come nucleosome. So round red mark is there. Red mark is covering by who are there? You know, double stranded DNA. So that's why dsDNA wrapped around the histone octamer is called as nucleosome. This nucleosome is a string-like structure, thread-like structure. So, but this the DNA is consisted how many base pairs are? 200 base pairs. And histone octamer is present. Octamer, art It means 8 molecules. So that's the histone octamer is consists 4 types of H2A, H2B, H3 and H4. Like each and every one which is produced another copies. So that's the 2 copies also. But fifth one is H1 histone is present. This green color marks, which is bind both of what is that mark? Double stranded DNA. So that's the fifth type of H1 histone seals the 2 tens of DNA. I think you're catching? Right, that's it. So like this, by the helping of DNA, which may connect it to another nucleosome. That's it. So that's the DNA that continues between the 2 successive nucleosomes are the that is called as linker DNA. The DNA by the helping of DNA only which may links to another nucleosome of DNA. That's it. So in our videos, molecular basis of inheritance, 5 questions I will give you given. I very clearly I have explained. So please watch it and subscribe my channel. This is the race tutorial. Guarantee one, sure 100% one essay kit will come. Very simplified, easily identified and clues are given. Please watch it and share it and subscribe. This is the race tutorial.